Question number 135. So, we are in respiration plants, fine. So, ATP is synthesized in that is like fifth complex, the electron transport chain that you see, FO, F1 particles, right. So, FO, F1 particles, where F1 particles which plays a very important role in the ATP synthesis. Okay, so option 135, 3 is the correct answer. That is F1 particle. 136, last electron acceptor in ETS, electron transport system, is oxygen. Oxygen is the correct answer for question number 136. Question number 137, out of 38 molecules of ATP produced upon aerobic respiration of glucose, the breakup in ATP production and glycolysis, pyruvate to acetyl-CoA formation Q and Krebs cycle R is as follows. So, when you say actually the ATP production and glycolysis, where pyruvate you could see 8 and then you could see Q, that is acetyl-CoA, that is 6, and R, that is a Krebs cycle, is a 24. So, 8, 6, 24, 8, 6, 24. This is P, you can say it is a Q, you can say it is a R. This is for this P that you can stand for glycolysis. This is for glycolysis. This is for acetyl-CoA, and this is for tricarboxylic acid acidic cycle. So, this is for the question number 137. So, you need to keep this thing in your mind, like where 2 pyruvic acid is formed, get the glycolysis. So, 2 pyruvic acid, 1 pyruvic acid and 2 pyruvic acid. Okay. So, you need to take in considerations for glycolysis where 2 pyruvic acid. So, P, Q is for acetyl-CoA, this is for a Q, and then you take for R part is a tricarboxylic acid cycle. So, we take P, Q and R. So, this comes like second option is the correct answer. So, P, Q and 137. So, we could see 1 glucose molecules in aerobic respiration is altogether what? It's 36 ATP. 36 ATPs we could see is out of 36 ATP, when we calculate 36 ATP is N10 NADH2 is equal to 30 ATP, then 2 FADH2, F2 FADH2 is equal to 4 ATP, okay, and 4 formed by substrate phosphorylation, you could 4 ATPs have been formed. So, you could see altogether 38 ATPs on the hood. Okay, so this gives actually 32. So, what happens, like here you could see 36 ATP, 10 NADPS, that is like 30 ATP, and 2 FADH2. 2 FADH2 gives to 4 ATP, and 4 this is actually like 4 formed by substrate phosphorylation. So, what happens in glycolysis and Krebs cycle, and 2 consumed in transport of NADH2. So, 2 is getting consumed. So, out of which what happened? This out of 38, this is minus 2 ATP. 2 ATP is consumed. So, you get 36 ATP on the hood. Okay, this is the consolidated part. We discussed already about this in a balance sheet when we discussed. Okay, so we move to the next part, 139. Acetyl coenzyme A can be formed through the process. That is 139. All the above: glycolysis, beta oxidation of fatty acids, the deamination of amino acids. Question 139, 4 is the correct answer. 138, you see during aerobic respiration, maximum ATP synthesized in, maximum ATP that you could see synthesized in the tricarboxylic acid cycle only. Okay. So, 138, 2 is the correct answer. So, 139 we already discussed. Next we move to 140, question number 140. When tripalmitin is used as a respiratory substrate, the value of RQ is less than 1, then tripalmitin you could see 0.7. This we already discussed for this one. Tripalmitin 140. Then 141 you take the respiratory quotient of glucose, the carbohydrates that you could see already 1.0. Then 142, consider the following statements with represent to the respiration. A, B, and D alone are correct actually. So, glycolysis occurs, the cytoplasm of the cell is correct. Aerobic respiration takes place within the mitochondria is correct, and D is C51H98O6 is the chemical formula for the tripalmitining of the fatty acid. So, A, B, D alone are correct. So, we are done with question number 142. Next we move on to 143. The number of oxygen molecules utilized in glycolysis. Number of oxygen molecules utilized in glycolysis is 0. is 0. Okay. So, option 1 is the correct answer for question number 143. Next, we move on to question number 144. If you look at the questions for question number 144, which enzyme helps in the transfer of the phosphate group from ATP to the carbohydrates? When you look at this question and look at this options where phosphatase, ATPase, phosphorylase, catalase. Option 3 is the correct answer, phosphorylase, because the transfer of the phosphate group from what? From ATP to a carbohydrate is refers to as phosphorylation and is carried out while in the presence of phosphorylase enzymes, that is glucose is phosphorylated. Example you take, glucose is phosphorylated to glucose 6-phosphate by ATP in the presence of enzyme hexokinase or glucokinase and magnesium ions. So, 144 question, option 3 is the correct answer. 145, glycolysis term as originated from Greek word, that is glycos and lysis. Option 2 is the correct answer. Question number 146, in glycolytic pathway, which of the following statements shows a reduction of coenzyme? Reduction of coenzyme glyceraldehyde 3-phosphate to 1,3-diphospho-glyceraldehyde. So, question number 146, option 3 is the correct answer. Question number 147, the end product of glycolysis. You know, the end product of glycolysis that I discussed already is the end product is pyruvic acid. So, pyruvic acid is the end product. So, this one for question 147, 1 is the correct answer. 148, in which of the one of the following process carbon dioxide is not released? Where? Lactate fermentation. Lactate fermentation is actually which occurs in the absence of oxygen. So, lactate fermentation. So, option 4 is the correct answer for question number 148, because anaerobic respiration of fermentation can be of two types, that is lactate fermentation and ethanol fermentation. Also, you take acetic acid fermentation. Lactate fermentation produces lactic acid only, as pyruvic acid produced in glycolysis is directly reduced to NaDH2 to form lactic acid and no carbon dioxide is produced, whereas you could see perhaps in ethanol fermentation, which involves a concentration of pyruvate to acetaldehyde, hence CO2 is released. So, 148, option 4 is the correct answer. Question number 149, the number of ATP produced when a number of molecules glucose undergoes fermentation. In case of fermentation, you could see only 2 ATPs. Question number 149. Question number 149, 2 ATP. Okay. Question number 150. Which statement is wrong for Krebs cycle? There is actually which statement wrong in a Krebs cycle. The cycle starts with the condensation of acetyl CoA, that is acetyl group, with pyruvic acid to yield citric acid. So, here you could see Krebs cycle which starts with the condensation of what? Condensation of acetyl CoA. Condensation of this acetyl CoA only, which occurs where the beginning which takes place, with oxyl acetate to form tricarboxylic and 3-carbon compound which is formed, that is citric acid. So, 150, option 3 is the correct answer. Question 151, connecting link between this is a glycolysis, this is a tricarboxylic acid. So, which is connecting this between acetyl CoA, which acts like a connecting, because the pyruvic acid you see is generated by the glycolysis, which enters into the mitochondrial matrix, undergoes oxidative decarboxylation and forms active acetate. Then that is acetyl CoA. The acetyl CoA is accepted by the Krebs cycle, therefore it acts like intermediate compound between glycolysis and the Krebs cycle. So, this acts like intermediate compound between these two, that is glycolysis and Krebs cycle. So, option 4 is the correct answer to question 151. 152, oxidative decarboxylation of pyruvic acid results in the formation of, acetyl CoA, we could see carbon dioxide, and also you could see NaDH2. So, option 4, that is 1, 2 and 4 only. Question 153, pyruvic acid, the key product of glycolysis, can have a many metabolic fates under aerobic condition. It forms acetyl CoA plus carbon dioxide. So, option 3 is the correct answer. So, for question 153, option 3 is the correct answer. Question number 154. 154, the TCA enzyme is found in, the TCA enzyme which found in, I told you, in mitochondrial matrix 154, mitochondrial matrix, the TCA enzyme which forms. Okay. So, 155, pyruvate dehydrogenase complex needed for the conversion of pyruvic acid to acetyl CoA is located in matrix of mitochondria, located in the matrix of mitochondria. So question 155, option 1 is the correct answer. Because the pyruvate, you see, the dehydrogenase is an enzyme that is needed for the conversion of pyruvate to acetyl-CoA. The enzyme is present where? In the mitochondrial. This present in the mitochondrial matrix. So this is for question number 155 also. 155 and 154. 156. Oxalosuccinic acid, an intermediate compound of Krebs cycle, is 6 carbon compound. 6 carbon compound, that is option 2. Option 2 is the correct answer. 157. 157. A small protein attached to the outer surface of the inner membrane and which acts as a mobile carrier for the transfer of electrons between the complex 3 and 4, between 3 and 4 with the cytochrome C. So between the complex 3 and between the complex 4, right, in between the complex which acts like something called a cytochrome. So cytochrome. So cytochrome which acts in between the complex 3 and 4. This is for the question number 157. Then question, so option 2 is the correct answer. Question number 158. During movement of electron through electron transport chain, pH of matrix increases. pH of matrix increases because the transport of electrons from NADH over the electron transport chain helps in the pushing 3 pairs of protons to the outer chamber, while the 2 pairs of protons are observed from the matrix during what? During the electron flow from FADH2. Then what happens? As a result, then this concentration of hydrogen ion in matrix reduces. Then what happens? Reduces proton concentration, results in increased basicity of the matrix, that is like, the pH of the matrix is getting increased. So for this question, option 1 is the correct answer. Question 159. Oxidation of one molecule of NADH2 give rise to, because one NADH2 I told you already. Question number 159. One NADH2 which gives rise to 3 ATP molecules, which gives rise to 3 ATP molecules. Okay. Question number 160. During anaerobic respiration, number of ATP molecules generated by the breakdown of 20 glucose molecule. So 20 glucose molecules, you can see it is for example 160. It is 40. It is 40 because aerobic respiration generates only 2 ATP. Anaerobic respiration which generates, I told you already. Anaerobic respirations which gives rise to only 2 ATPs, right? 2 ATPs per glucose molecule. So on the breakdown of 20 glucose molecules, then what happened? 20 into 2. So you need to multiply this 20 into 2. So this which gives rise to 40 in number. So for this question, 160, 40 is the correct answer. Question number 161. How many ATP molecules will be generated in the plant system during complete oxidation of 40 moles of glucose? So complete oxidation, the calculation if you do is like 1520. So correct option is 1520 for question number 161. 162. The respiratory quotient value of 0.7. 0.7, this we already discussed. Tripalmitin. 162. 162 tripalmitin. Tripalmitin is 0.7. That is less than 1 for question number 162. Next we move on to the next, that is next question 163. 16. So we move on to the next question 163. Move on to the next question 163. Select the wrong statement. If you look at the 4 options, you see the second option which seems to be incorrect. The intermediate compound which links the glycolysis with Krebs cycle is Malik cycle. It is not a Malik cycle, it is acetyl-CoA. So option 2 is the incorrect answer. 164. The respiratory quotient during cellular respirations would depend on the nature of a substrate. The nature of a substance. So option 4 is the correct answer for question number 164. 164, option 4 is the correct answer. Next we move on to question number 165. Assertion and reason. Conversion of 1,3-bisphosphate to 3-phosphoglyceric acid PGA in an energy yield step. And reason, this energy is trapped in the form of ATP. Both assertion and reason are true, but reason is not the correct explanation of the assertion for question 165. This is because of one of the true phosphates of 1,3-bisphosphoglycerate, that is BPGA, we call it as linked by high energy bond. It can synthesize ATP and form 3-phosphoglycerate by Phosphoglycerate kinase, that is an enzyme. In this, the direct synthesis of ATP from the metabolite is called substrate level phosphorylation. This we already discussed before the first part. So 165, option 2 is the correct answer. Option 160, question number 166. In which of the following reactions of glycolysis oxidation takes place? 3-phosphoglycerol degrade through 1,3-bisphosphate. So we discussed in the beginning of the first part. So 166, option 4 is the correct answer. 167, the net gain of ATP from one molecule of glucose of glycolysis is only, the net gain is only, the gain is only 2 ATP, because glycolysis we call it as Empron-Mayer pathway, the other name is a series of biochemical reactions in which glucose is getting broken down into pyruvate with the release of usable energy in the form of ATP. One molecule of glucose undergoes 2 phosphorylation reactions and is then split into 2 triose phosphate molecules. Each of this is getting converted to pyruvate. The net energy yield is 2 molecules per glucose. Okay. So option 4 is the correct answer. Question number 168. Conversion of pyruvic acid into ethyl alcohol, that is decarboxylase and dehydrogenase. Decarboxylase and dehydrogenase. For question 168, option 4 is the correct answer. Question number 169. Coming to the 169. That is, select the suitable name for the following process in case of 169. This is a kind of fermentation where you could see the glucose molecule which gets combined with 2 ADP and 2 phosphate and leads to the formation of ethyl alcohol. Since ethyl alcohol is getting formed, this definitely it must be alcoholic fermentation. So this is not aerobic fermentation. So option 1 is the correct answer for question number 169, because the end product that you get is ethyl alcohol, then this must be alcoholic fermentation in the absence of oxygen. Question number 170. 170, the energy releasing metabolic process in which substrate is oxidized without an external electron acceptor is fermentation. It is a fermentation. That is question 170, option 2 is the correct answer. 171, in single Krebs cycle the decarboxylation takes place. Decarboxylation takes place in what? In 2 steps actually. One step is what? One step, the oxalosuccinate. The oxalosuccinate, where it is getting converted to alpha ketoglutarate, and then what happened, the alpha ketoglutarate, that is 5 carbon compound which combined with CoA, and by with the help of enzyme alpha ketoglutarate, this will converted to succinyl CoA. So that is succinyl CoA is a 4 carbon compound. So 6 carbon compound is getting converted to 5 carbon compound, and 5 carbon compound eventually is getting converted to 4 carbon compound, that is succinyl CoA. So you could see 2 steps which involved in this process. Question number 172. In 172, FAD is an electron acceptor in the citric acid cycle during the oxidation of succinic acid. So what happened? The succinic acid is getting converted to the malic acid. So succinic acid is getting converted to malic acid for question number 172 by means of enzyme succinate dehydrogenase. Question number 173. During aerobic respiration, acetyl CoA is synthesized in mitochondrial matrix. This we already discussed for question number 173 also. Okay, 174. Match the organic compounds listed under the column 1 and 2 and choose appropriate from the given choices. When you look into this, where A is equivalent to T and B is for S and C is for R and D is for Q. For D you could see acetyl coenzyme, it's a 2 carbon compound, whereas like phosphoenol pyruvate you could see it is a 3 carbon compound. So whether it is 2 carbon compound, 3 or 4, 5 carbon compound, so this type area also you can get a question. So you can be extra cautious when you attempting for this type of question. If you can answer for any 2 options correctly, then you can answer the rest of the things as well. So option 1 is the correct answer. Question number 175. TCA cycle is named after. So this person, the Hans Krebs, he is the first person who first identified the tricarboxylic acid cycle. So we call this cycle as a tricarboxylic acid cycle. So option 175, 2 is the correct answer. Question number 176. In citric acid cycle, the step which is not mediated by dehydrogenase enzyme, is oxaloacetic acid to the citric acid. Where citric acid that we discussed just now, where this can be also called as Krebs cycle or citric acid cycle, because the first stable compound which formed in this cycle is citric acid. So, in Krebs cycle, it's stepwise oxidative and cyclic degradation of acetyl-CoA derived from pyruvate. Dehydrogenase enzymes mediate the conversion of citric acid to alpha ketogluteric acid, succinic acid to fumaric acid, and malic acid to oxaloacetic acid. So, what happens? Oxaloacetic acetate converts into citrate in the presence of citrate synthase. So, option 176, 1 is the correct answer. We move on to the next question, 177. Which one of the following reaction is an example of oxidative decarboxylation? In oxidative decarboxylation, you could see conversion of pyruvate to acetyl-CoA, where oxidative decarboxylation is the link that I told you between the glycolysis and the tricarboxylic acid cycle, where you could see in this phase the pyruvate which is formed in the cytoplasm by glycolysis produce CO2, NADH2 and acetyl-CoA by oxidative decarboxylation reaction. Acetyl-CoA functions as substrate that is entrant for the Krebs cycle. Okay, so question 177, option 3 is the correct answer. Question 178, FAD act as an electron acceptor in between, where you could see this we discussed, in between the succinic acid and the fumaric acid, where you could see in this case the succinic acid which involves in fumaric acid, the enzyme involved in succinic acid dehydrogenase. This is the one reaction of Krebs cycle which does not involve the utilization of cofactor, that is NAD or FAD, and that is NAD or NADP, and the place by which taken by that is FAD, flavin adenine dinucleotide of the enzyme, succinic acid dehydrogenase. This FAD takes place of the 2 hydrogen ions, and then what happens? The 2 electrons from succinic acid and it gets reduced. So, for question 178, option 2 is the correct answer. Question 179, where enzymes of TCA cycle are present. So, where the enzymes of TCA cycle, this is already discussed many times, where the enzymes of TCA cycle for question number 179 also, usually this present in the mitochondrial matrix, there we call it as mitochondria also. So, the Krebs cycle is also named as citric acid cycle or the tricarboxylic acid cycle. After the initial product, the Krebs cycle is a stepwise oxidative and cyclic degradation of activated acetate derived from pyruvate. So, it occurs inside the matrix of mitochondria because enzymes of TCA cycle are placed in the mitochondria. So, for this question 179, mitochondria is the correct answer. Question number, that is the last question for this chapter. During one Krebs cycle, number of carbon dioxide molecule released. So, this we discussed how many like carbon dioxide, because when we talk about 2 Krebs cycle here, tricarboxylic acid cycle that we discussed, in each Krebs cycle you could see 2 molecules of the carbon dioxide which is getting liberated outside. So, during one Krebs cycle what happens, the number of carbon dioxide released is 2 is the correct answer for this questions. By this we come to the end of this particular chapter, that is 180. The question for 180 of correct option is 2. 2 is the correct option. So, we are done with this chapter, the respiration plants. So, guys, when we talking about this chapter, we would have seen like out of this questions, so many questions, the repeated questions been asked from the glycolysis, then the tricarboxylic acid cycle, and again the questions which has been asked from the NADPH to ATPs and everything, and where this especially the process takes place. So, these are the areas that you should focus. So, by this we come to the end of this particular chapter, that is the respiration in plants. But still, if any queries or doubts in this part, you don't please hesitate to ask me. Thank you so much.